Unless you've been living under a rock, you've likely heard about CRISPR-Cas9, a technology which allows us to literally edit DNA, but also brings with it some ethical concerns and has sparked debate about whether or not we even have the right to edit genes. But how does this perhaps revolutionary technology function? And where does it even come from? In nature, CRISPR is actually part of the immune system of some species of bacteria, and it's used to fight against viruses. The way it works is that the bacterial proteins Cas1 and Cas2 will find a sequence of the invading virus's DNA, rebuild it, then insert it into a CRISPR array, which is an array of clustered, regularly interspaced, short palindromic repeats within the bacterium's genome. I like to think of this process as sort of like copying a new phone number into a phone book. Because the phone number, which is the viral DNA sequence, is now stored in the phone book, which is the CRISPR array, the bacteria can easily use it whenever it needs to. But when would it need to? If the bacterium is again invaded by a virus, which is very likely, RNA transcripts of the sequences, which are now accessible in the CRISPR phone book, will be paired with Cas9 proteins, which will then cleave the virus's DNA at the very specific spots that the RNA transcripts correspond with, which will hopefully stop the infection. So now that you understand naturally occurring CRISPR editing, you might be able to understand how our CRISPR-Cas9 technology works. By pairing it with engineered sequences, we can exploit the Cas9 protein so that it can be used to cut sequences of DNA that we want it to. Thus, we can edit genomes by injecting these Cas9 enzymes into cells, thereby cleaving unwanted genes. In addition, we can then manipulate the cells to perform specific gene repairs, allowing us to insert wanted genes. This engineered destruction and insertion of specific genes is how we can cure genetic disorders. For example, sickle cell anemia, a fairly common genetic disease, has already been treated using CRISPR-Cas9. As CRISPR-Cas9 gets more accessible and cheaper, we might gain the power to change millions of lives. However, just like with any novel technology, there's a chance of it being misused or even weaponized. And because of this, we have to actively make sure that those millions of lives are changed for the better and not for the worse.